Well, good afternoon, everyone. Good afternoon and good evening. And hopefully not a very good early morning to some regions where you're joining in from. Very excited to be joining this kind of an unorthodox session from Axiom itself. As you know, we are a think tank based in Princeton, Boston in India, as well as our setting in Europe. We are here today to kind of walk you through some of the trends that we have been evaluating as a think tank ourselves. That's kind of what we do in terms of trend spotting. The team is very excited to present some of their initial understanding of how 2021 rolled out and what are some of the trends we are seeing and excited about in 2022. Quickly walking you through, today I'll be joined by my co-host here, Dr. Mahim Misra, and we'll be walking you through myeloma, melanoma, lung, breast, hematology, GI, prostate, bladder, RCC, as well as, very interestingly, a pan-tumor approach of precision medicine. With that, Mahim, what do we have planned for today?
Awesome. Thank you so much, Hafiz. So we'll go through each of these tumor types that Hafiz mentioned one by one, and I'll invite our speakers as we move along. But to start the discussion, I wanted to highlight some of the key themes that you'll hear across different tumor types. So starting with checkpoint inhibitors, they have been transformative in metastatic setting across different tumor types, and now we have seen them starting to move into earlier lines of therapy. And there is a strong case to be really optimistic about some of these checkpoint inhibitors movement to early stage setting, leading to increased survival outcomes. In terms of second theme is around antibody drug conjugates, which I'm sure everyone on the panel as well as in the attendees must have heard about it. It now have increasing role in treatment paradigm across different tumor types. Of course, breast cancer was one of the first tumor types where ADCs started to show increased efficacy. And now we are also seeing either approvals in late line setting, as well as a lot of phase 3 first line trials across different tumor types, especially lung cancer and bladder cancer. Immuno-oncology have been dominated by PD-1, PD-L1 inhibitors, as well as CTLA-4 inhibitors. And this field has been waiting for some more transformative mechanism of actions to enter market. And 2022 will be that year when some novel IO MOAs will enter the market either in combination with or without checkpoint inhibitors. Some of the MOAs to highlight would be TIGIT, particularly in lung, where we might see a phase 3 data readout in PD-L1 positive first-line non-small cell lung cancer. There is a LAG-3 data which already read out and could enter market in melanoma in combination with a PD-1 inhibitor. And then, of course, novel cytokines, especially IL-2, could have phase 3 data readout, pivotal data readout across different tumor types, as we highlighted here, including lung, melanoma, and RCC. So really a critical year to see how some of these MOAs, essentially the second wave of immuno-oncology, could be effective in changing the treatment paradigm. Last but not the least, there has been increased use of liquid biopsy, not just for selecting patients, but also for monitoring patients in different tumor types. It has been in heme malignancy, particularly as part of as MRD has been part of clinical decision making more and more now. But we are also seeing the utility of liquid biopsy, not just in late setting, but also in early stage setting, in specifically in tumor types like bladder cancer and GI CRC. And we'll talk about it when we move to other tumor types. What is not mentioned here, but I'll be remiss if I don't talk about it, would be bispecifics. We are starting to see that bispecifics has started to regain momentum with few approvals in lung and an expected approval in melanoma, which we'll talk about as well. And then, of course, cell therapy, heme malignancy have been the poster child of cell therapy, especially autologous cell therapy. And they have been having hurdles to move to solid tumors there, but there are some early signals that new modalities like TILs or allogenic cell therapy could help cell therapy to move into solid tumors as well, with some of the validated targets in different tumor types. With that, I'll pass it back to Hafiz to introduce us with the list of speakers.
Great, and thank you, Mahim, for the point of view. That is super exciting. And as I had mentioned in my narration on LinkedIn and other places as well, we are open to your comments, your questions, as well as your hypotheses. Feel free to kind of push us in terms of our thinking. These are our point of view, and happy to address some of the questions through the chat, as well as the Q&A avenue of the Zoom as well as LinkedIn. With that, we'll jump right in, and Dr. Andrew Hong is going to take us through lung cancer and where has that been in 2021 and what are we excited about in 2022.
Thanks, Hafiz. 2021 was an exciting year for lung, and here I'll be introducing key themes that we'll see my colleagues build upon in other indications. So we'd like to start off with targeted therapies, where it's important now more than ever to conduct biomarker testing. In May 2021, we saw the FDA approve sotorasib, making it the first commercial treatment for lung patients with a KRAS G12C mutation. KRAS G12C was deemed a historically undruggable target. And shifting over to antibody drug conjugates, or ADCs, as Mahim alluded to, ADCs are on the move in lung, with HER2 data presented last year. As reference, no ADCs are currently approved in lung, though my colleagues will share their role in other solid tumors. Spotlighting novel IO, just last month at ESMO IO, Roche shared data for atezo plus tiragolumab, which is a TIGIT inhibitor. Notably, overall survival was not reached in the PD-L1 high population. Questions remain about the role of TIGIT in all comers, especially in an increasingly competitive space. Looking forward to 2022 promises to be another important year in lung, with potentially practice-changing data sets on the way. Here, we'll discuss a few anticipated events across targeted therapies, checkpoint inhibitors, and novel IO. For targeted therapies, we saw just a few weeks ago sotorasib approval in the EU for the same launch indication as in the US. That said, it's still early days for the KRAS G12C class. Expect data from adagrasib, another KRAS G12C inhibitor, later this year, with its potential approval on the horizon. We're excited to see more data sets for KRAS G12C inhibitors in brain metastases and in combinations with other modalities such as PD-1, SHP2, and EGFR. Shifting gears to checkpoint inhibitors, we won't need to remind this audience, but PD-1-based therapies are the standard of care in first line. What we're seeing now is the arrival of other PD-1s like Lilly's sintilimab hailing from China. This will be up for approval coming March, and it's a watershed moment for the FDA, potentially opening the door for the other PD-1s with downstream implications for the field. Keeping with checkpoint inhibitors, another theme is PD-1s moving into earlier lines of therapy, particularly beyond the metastatic and into the neoadjuvant and adjuvant spaces. My colleagues will further explore this trend in melanoma, RCC, bladder, breast, and GI. In recent memory, we've seen this trend for both checkpoint inhibitors and for targeted therapies. So recall the approval of atezo in adjuvant lung and osimertinib in adjuvant EGFR based on ADAURA. Both use DFS endpoints for approval, and in the future, we're looking for additional data from the combo of nivo and ipi and neoadjuvant to shed light on the potential role of IO-IO combinations. For novel MOAs, the TIGIT narrative continues to shape up, with data on the way for first-line lung to helping characterize the role of TIGIT beyond PD-L1 high only. So looking broadly at lung, it's important to note that the 5-year survival rate has improved, especially with the advent of checkpoint inhibitor-based therapy. For ADCs, we may see the first class approvals in lung this year, and with additional targeted therapies approved and even more on the way, the need for testing continues to be paramount. Stay tuned for novel MOAs on the horizon, like TIGIT, and with that, I'd like to open it up for questions.
Great, and as we mentioned, please send in your questions for the panelists as well as the speakers in the forum today. So Andrew, as we warm up with some of the questions that are coming in, do you feel that in NSCLC, is NSCLC becoming more accepting of other endpoints beyond overall survival?
It's a great question. And the short answer is yes. So the field is shifting away from accepting only positive OS data from phase 3 trials. You see several examples of this over the past year with sotorasib and amivantamab, both approved accelerated approval on phase 1-2 trials. And if you remember the ADAURA debate for osimertinib, approval was based on DFS then, and now we're seeing that adjuvant atezo was approved based on DFS as well. So we'll still need to see longer follow-up to see if the OS proves positive, but KOLs are asking, is there any reason why these drugs shouldn't be available, especially in the case of osimertinib, where you see activity in brain mets? So the open question now is, do we have to expect OS improvement? Has the bar shifted in lung? And this will continue to be debated in years to come.
Awesome. That is very interesting and something we'll definitely track in terms of how endpoints evolve within lung cancer, especially in early stage setting. Andrew, you mentioned about antibody drug conjugates and potentially they might enter in lung cancer. Can you elaborate a little bit what are different targets being evaluated within ADC? What does it mean in treatment paradigm?
Yeah, gladly. So taking a step back, ADCs have already shifted the practice of treatment in other indications like breast cancer and bladder cancer, for example. But just to remind the audience, ADCs are not currently approved in lung. So what we're seeing now is there are multiple ADCs with encouraging data, like trastuzumab deruxtecan in HER2 positive lung, and sacituzumab govitecan, which is a TROP2 targeting ADC. Just earlier this month, there was a phase 3 trial for sacituzumab in combination with pembro in first-line lung. And time will tell how ADCs shape the treatment of lung, but the class is here to stay, and it's worth keeping a close eye on moving forward.
Awesome. That is great. And again, I just want to reiterate, if any of the attendees have any questions for one of our panelists, please feel free to use the Q&A feature on the Zoom webinar link. We'll be happy to take your question.
Great. So in the interest of time, moving right along, we have our next panel, Dr. Ann Cherry, who will be walking us through melanoma. How was the past in 2021 and what are we excited about in 2022?
Thank you, Reese. Thank you, Mahim. This has been an impressive year for melanoma, starting off with the data shared at ASCO from Relativity 047, showing a PFS benefit for patients with first-line melanoma from a combination of PD-L1 therapy and LAG-3 compared to nivo monotherapy. And this was exciting because it was the first clinical validation for a checkpoint other than a PD-1 or CTLA. So it's exciting to see continued innovation in immunotherapy within the melanoma space. And of course, one of the ongoing challenges in melanoma has been determining the best sequencing strategy for patients who are eligible for more than one therapeutic option. And in September, the SECONDA trial brought us one step closer to a data-supported standard of care for these patients by demonstrating that BRAF patients benefit from receiving their TKIs either after IO or as a sandwich style before and after. And then finally, 2021 wrapped up with an exciting ESMO and rapid FDA approval of Merck's data demonstrating that pembrolizumab could reduce the risk of relapse in patients with quite early stage disease, stage 2B and stage 2C melanoma, with hazard ratio of 0.65. And the success of this trial speaks to the power of immunotherapy across the patient journey, even for those who are initially diagnosed with relatively early stage disease, but who may have a relapse later on. So coming into 2022, a few of the things that we're watching this year, we're expecting to see approval of the LAG-3-nivo combo that was presented at ASCO last year, as well as EU approval of pembro in stage 2 disease. Nivolumab is also hard on the heels in stage 2 with Checkmate 76K data should be available in the first half of this year. There are also some new approaches that we're keeping an eye on that are exciting. The most recent example of melanoma as a proving ground for novel IO, LEAP-003 is evaluating checkpoint inhibition in combination with the novel TKI lenvatinib, and PIVOT-IO is testing nivolumab and BENPEG, the innovative IL-2 based therapy. And then, of course, last thing to mention here, we're excited to see progress from Iovance's tumor-infiltrating lymphocyte therapy, which has faced some regulatory challenges, but delivered very impressive responses for hard-to-treat melanoma patients. And this BLA should be submitted within the next few months. So headed into this year, we're expected to see some of the trends of continued solidification of the role of PD-1s, including these innovative combinations and expansion to new patient populations within melanoma. We also see that multiple manufacturers are working to determine the best way to co-administer TKIs and PD-1s for BRAF patients, whether that's simultaneous triplet administration or sort of more on the sequencing question. And then finally, while, of course, PD-1 therapy has revolutionized melanoma care, not everyone responds. And of course, most patients do eventually relapse. So we're excited to see new and creative approaches here, like Iovance's TILs, take on the challenge of some of these hard-to-treat patient populations. So let's see if there are any questions coming in from the audience.
Great, and you kind of alluded to that melanoma being a bellwether, correct? So that being a bellwether in the forefront of IO, how does the progress here predict what we might see in other tumors coming in the next few years?
That's a really good point, Hafiz. And in many ways, melanoma has been the proving ground for IO therapy. It's one of the original tumors where this was evaluated and really brought to success for the first time. And I think there's really two things that I would keep in mind here and I would keep an eye on. One is we're going to see this continue in the testing of new combinations of PD-1s with new MOAs. I think we're going to see a lot of activity in melanoma trying to come up with what is the new and better combination for both first line and later line populations. And I know that some of our thinkers from lung and bladder, and I believe RCC, are going to be talking about this in about 20 minutes. And then the second one is the movement of PD-1s to an earlier patient population, particularly adjuvant therapy, and helping prevent those patients from relapsing who otherwise would have relapsed potentially with very severe metastatic disease down the road. And one of the things that we're going to be keeping an eye on is whether the all-comers approach that worked in melanoma, everyone with stage 2 or stage 3 disease, was treated with adjuvant therapy here, and overall there was a significant benefit. Whether that all-comers approach also holds for other tumor types, or whether other tumors are going to see more, are going to need to stratify the patient population a little bit more, potentially by risk of relapse, in order to see checkpoints move into adjuvant.
Great, thank you, Anne.
Thank you so much. There is one more question. It's a great question, but what do you think are the remaining unmet need in melanoma, Anne?
Well, until recently, I would have said uveal melanoma. This is an area of melanoma that has been historically left behind. But with Immunocore's recent data, 0.51 hazard ratio of overall survival for uveal melanoma, it seems likely that this group is going to have at least one solid option starting this year. And it's a very exciting first CD3 bispecific, not only in melanoma, but in any tumor type. Which means that the ongoing unmet need here is going to be that post-PD-1 population. And that's a mixed group of patients who never responded when they received checkpoint inhibitors, those who were initially refractory, as well as those who have a good response for a number of years, but then do eventually relapse.
Awesome. All right. Thank you. Thank you so much, Anne.
Okay. Great. So now we have Jamie Tanzer, who will walk us through RCC. Jamie is joining in from Boston.
Thanks, Hafiz. So this past year, RCC has held several key events, ranging from providing marginal benefits to existing approaches to more groundbreaking readouts and approvals. When 2021 started, the frontline standard of care was mostly made up of IO combinations with TKI monotherapy as the favored second-line standard of care. And this year, we saw additions to that frontline IO combination set of regimens with the approval of nivolumab and cabozantinib based on the CheckMate 9ER trial, which provided stronger efficacy data than had been seen to date, and started the debate around the role of quality of life analyses and treatment selection, as this trial was the first IO-TKI combination to demonstrate a quality of life benefit over the control arm. This was quickly followed by the data release and later approval of pembrolizumab and lenvatinib based on the CLEAR trial. This study set a new standard for IO-TKI efficacy data, particularly for the complete response rate. Both of these approvals contributed to the continued domination of IO combinations in the frontline setting. On a slightly different note, we also saw exciting news with Merck's HIF-2α inhibitor belzutifan becoming the first approved treatment for von Hippel-Lindau disease, for which one of the symptoms is RCC tumors. This bodes well for the potential role of HIF-2α inhibition in the broader RCC space as well, and HIF-2α is currently considered by many to be the most exciting novel mechanism for RCC. And finally, after more than a decade of adjuvant trials that failed to demonstrate clinically meaningful benefit, this year we saw data and FDA approval of pembrolizumab as a groundbreaking new option for the treatment of patients with localized RCC at intermediate, high, or high risk of recurrence. In line with what we've seen in other solid tumors, this is another example of IO being moved earlier in the treatment paradigm. It's yet to be seen whether moving IO earlier in this case will translate into a long-term OS benefit, although a preliminary trend towards survival benefit was seen in the initial data readout. Now looking forward to 2022, in the adjuvant setting, we expect to see Emotion 010 readout data on atezolizumab. So based on relatively weak data on atezo and other RCC trials compared to pembro and nivo, it's hard to say what kind of data we should expect to see here. We also might see data from Checkmate 914, testing a more aggressive approach of PD-1 plus CTLA-4 rather than PD-1 monotherapy. This could enhance response rate and potential to cure patients after surgery, but it could also come along with severe and permanent adverse events. Both of these trials will be valuable in helping to elucidate the role of IO in the adjuvant setting, following the approval and initial use of pembro. Now, in the metastatic setting, we're expecting to see readout of the final pivotal doublet trial, PIVOT-09, which is looking at nivo plus bempeg in the frontline setting. For reference, bempeg is an IL-2 prodrug that aims to provide more targeted effects on the tumor without the systemic toxicity seen with high-dose IL-2 in RCC years back when that was the standard of care. And finally, COSMIC-313 will bring us into the triplet realm, an intriguing new frontier for RCC that aims to further enhance efficacy outcomes. This trial assesses nivo plus ipi plus cabo, and there are high hopes for the complete response rate and durability of response, although there are also concerns around toxicity. So now thinking about some trends and implications, we've seen IO combinations now almost entirely displace TKI monotherapy in the frontline, which is currently hyper-competitive with 4 IO-TKI combinations and 1 dual IO combination as standard of care regimens. We also see IO entering the adjuvant setting, and even in the next couple of years, we'll expect to see some data on IO retreatment in the second-line plus setting. Circling back to the frontline, we see a trend of looking at triplets to reach the next level of efficacy, including with COSMIC-313 as mentioned on the prior slide. Another strategy to improve efficacy is through investigating biomarkers and molecular drivers to tailor treatment to each patient. PD-1 has historically not been a useful predictive biomarker for RCC, so we're seeing a continuing search for more useful biomarkers to assist in treatment selection, including in the adjuvant setting. And as somatic VHL mutation is a driver of disease for many clear cell RCC patients, HIF2 alpha inhibitors in particular are a source of much anticipation. So at this point, I would love to address any questions. Oh.
Thank you so much, Jamie. So in your presentation, you mentioned how a new novel cytokine, bempeg, is going to get, would have a data readout in 2022. Can you elaborate a little bit on where you see, what you see the role of these novel cytokines in RCC?
Sure. So in RCC, as we touched on a little bit throughout the presentation, there are really 2 main avenues that are being addressed, which is immunomodulation and anti-angiogenesis. And so the nivo-bempeg combination fits more into the immunomodulation pathway. This is something that, as I mentioned, has been investigated for years and years. Some very early therapies in RCC were more in the cytokine realm. And with bempeg, we've seen some initial data coming out from earlier phase trials in RCC as well as in melanoma and in bladder. And there is some anticipation that the prodrug could provide more targeted benefit with less systemic toxicity. So it'll be interesting to see whether this can contribute to durable responses, but we'll have to see what the data shows this coming year.
No, absolutely. Absolutely.
Great. Okay. With that, thank you, Jamie. We'll move on to Bladder. And we have Sarah, who's joining you from Princeton, taking us through data in Bladder.
Hi, thank you, Hafiz. So I have the opportunity to discuss some of the major developments in bladder cancer over 2021 and what they might mean for the year ahead. So starting with the metastatic setting, in the first quarter of 2021, the second-line post-chemo indications for atezo and durvalumab were removed based on the lack of confirmatory phase 3 evidence. Both of these agents were previously granted accelerated approval based on phase 2 studies with overall response rate endpoints. However, because no confirmatory data trial has been able to demonstrate survival benefit in this setting for either agent, the indication was removed from the labels and both were removed from NCCN guidelines for bladder cancer. Looking towards later line settings, 2021 also brought about the approvals of ADCs. enfortumab vedotin, which is a Nectin-4 targeting ADC, was previously granted accelerated approval, but in 2021, the FDA granted it full approval based on OS benefit seen from the EV301 study post-chemo, post-checkpoint inhibitor. Sacituzumab govitecan, which is a TROP2 targeting ADC, was granted accelerated approval based on the TROPHY-U01 study. Shifting to the non-muscle invasive bladder cancer setting, Vicinium, an intravesical agent investigated for BCG unresponsive non-muscle invasive bladder cancer, received a CRL by the FDA. The trial that was intended to be registrational for Vicinium was the VISTA study, and the CRL cited manufacturing and data quality issues that needed to be addressed. One of the biggest changes to the landscape was the approval of the first immunotherapy within muscle invasive bladder cancer. Nivolumab was granted FDA approval as an adjuvant therapy based on DFS data from the phase 3 CheckMate 274 study. This approval is a big step forward for muscle invasive bladder cancer, but it remains to be seen how physicians will use adjuvant nivolumab in the absences of OS data. As Andrew discussed in lung cancer, the role of DFS and the potential necessity of OS is still up for debate. On the next slide, we have some potential data and approvals that might be expected in 2022. So at ASCO GU 2022, we're expecting to see data of N803 plus BCG for BCG unresponsive non-muscle invasive bladder cancer patients. This is an IL-15 agent that is administered intravesically. And we've already seen some preliminary positive data for this combination. This data may serve for the basis of regulatory application and may lead to approvals in the second half of 2022. Also within the BCG unresponsive non-muscle invasive bladder cancer setting, there may be an approval of an intravesical agent adstiladrin that demonstrated promising efficacy, but was given a CRL based on manufacturing challenges back in May of 2020. If the manufacturing challenges can be addressed, we may see a potential approval this year. In the metastatic setting, based on recent CHMP positive opinion on EV301, we anticipate EMA approval for enfortumab vedotin for those patients who have previously received both the checkpoint inhibitor and chemo. This would be the first approval of enfortumab vedotin in the EU. Finally, looking at IMvigor130, data was originally presented at ESMO 2019, and this year we're hopeful to see the final OS data that might provide some additional insight into the role of IO plus chemo for first-line bladder cancer. Looking towards some of the key trends, we see a growing interest in ADCs, as ADCs are currently approved and are also being investigated in clinical trials in earlier lines of therapy. IO is also moving to earlier lines of therapy, especially with the approval of adjuvant nivolumab for muscle-invasive bladder cancer. It will also be interesting to track how the introduction of IO in earlier lines might impact treatment decisions in later lines. Finally, we're seeing some challenges for intravesical therapies in non-muscle invasive bladder cancer. Currently, Pembro is the primary agent approved for BCG unresponsive non-muscle invasive bladder cancer, and intravesical agents have faced manufacturing challenges that have acted as barriers to entry in the market. Thank you for your attention. I'd be happy to open it up to questions now.
Great. Thank you so much, Sarah. That was really really interesting. You mentioned how checkpoint inhibitor are entering muscle invasive bladder cancer. Do you expect any challenges once checkpoint inhibitor enters muscle invasive setting?
Absolutely. So there are a couple of things to keep in mind with the muscle invasive setting. The first is that it's very multidisciplinary. So within muscle invasive bladder cancer, both urologists and medical oncologists are part of the decision-making here. So really incorporating IO adds to that dynamic, and understanding how urologists are going to play a role in the use and uptake of IO in combination with the urologist is very important. Also understanding the potential role of the DFS endpoint, as I mentioned, and Andrew also mentioned, that might be a challenge to determine which patients might be most appropriate. So if you look at NCCN guidelines, it notes that it's for patients who prefer disease progression, but with unknown potential survival benefit. So identifying the appropriate patients to give IO to, especially in the adjuvant setting where it might be considered overtreatment. These patients have undergone a radical cystectomy and they no longer have a bladder. So how do you determine which patients should receive an adjuvant therapy?
Great, thank you. So, Sarah, in terms of, we hear about ADC around the table, around the shop quite a bit, right? So is this a trend, or what is the potential of ADCs in the future in this segment?
it's really, really exciting, the type of data that we're seeing for ADCs in bladder cancer. So enfortumab vedotin and sacituzumab govitecan have had really exciting data in the later lines of setting. But I think the future of it and where it's going is moving to first line. I think we've seen some initial data of Pembro plus EV in the first line, small n size, so just preliminary. But there seems to be a lot of enthusiasm for this combination. And if it can move to earlier lines of therapy, it might play a much bigger role within bladder cancer.
Sounds great.
Great, this is exciting. And so moving right along, shall we move on to breast? Mihir?
Oh, wait a second. I think we have few more questions for for our previous panelists. So I would like to invite Sarah, Jamie, and Andrew back to discuss some of the themes that cuts across their tumor types.
Okay, sounds good.
All right, so let me just start by posing a more broad question for everyone, and then I'll pose a specific question to Andrew to just get the ball rolling. So from your perspective, are there any new MOAs or targets that you think are exciting and have shown some proof of concept data in phase 1-2 that gives you more confidence in its potential in your tumor type? So let's just start with Andrew. I'm very curious about KRAS G12C inhibitors and what does it mean for lung cancer, and why was it considered such a big breakthrough? And any other MOAs or modalities that you would like to add to the growing list of high potential targets?
Yeah, absolutely, Mahim. So KRAS G12C, KRAS in general has long been held as an undruggable target. And it wasn't until the seminal paper from Shoka and Ostrom that they were actually able to drug this. And it's really important because of the prevalence. KRAS G12C is mutated in roughly 13% of adenocarcinomas with a preponderance for smoking. And this is a patient population with really high unmet need. What we're seeing now, I want to reiterate, is early days for the KRAS G12C class. You're seeing monotherapy data in a second line plus lung. Looking forward, there's open questions as to how this narrative unfolds, whether it's into first line lung, whether it's into other indications such as CRC and other solid tumors. And it's going to be really important to keep an eye out on combinations.
Awesome. So there is a question from an attendee, and I think it's for the bladder. So Sarah, the question is, what are your thoughts for the cohort K for EV1033 and what this could mean for the landscape?
Yeah, that's a really great question. So that's we've seen some preliminary data. So EV103 cohort K is EV plus Pembro as well. And so that there's a lot of dynamics to be incorporated here. So it's moving IO plus EV to earlier lines of therapy. And while that's really exciting and encouraging, bladder cancer right now is largely IO and ADC treatments. So pushing that further and pushing that combination up front kind of leaves the question, what do you do after that? If patients progress, what other options are there? How long before you retreat? So while it's really, really encouraging to have these options for patients that, you know, extend that potentially extend life, that have really strong response rate data, it leads to a lot of additional questions of what do we do after that and what other options are there, because ADC and IO are kind of it right now, other than chemotherapy.
Awesome. Thank you so much, Sarah. So Jamie, a similar question to you as well. Maybe if you can give a quick overview of why HIF2 alpha inhibitors were such a breakthrough within RCC, and then maybe give us a snapshot of where you think are the new targets or agents coming up in RCC.
Yeah, for sure. I mean, I'm definitely hearing a lot of parallels to what we just heard from my colleagues Andrew and Sarah with regards to this kind of exciting breakthrough on a target previously thought to be undruggable. That's kind of the same experience we've had with HIF2 alpha, as the target is a transcription factor. It was, not thought to be something we could target, but over the past few years, we have seen that innovation. And as I mentioned earlier, this is really an underlying disease driver for a pretty substantial chunk of patients. So really exciting news there. In terms of other mechanisms that we're seeing, one that comes to mind is adenosine inhibition. So this is kind of a new approach that's being investigated to see how it could play into immunomodulation, particularly with, as Sarah was mentioning, Similar to in bladder, in RCC, there's kind of a limited set of mechanisms or drugs that we have. So in addition to looking into PD-1 retreatment, there's also interest in seeing like what other elements of immunomodulation could we potentially bring into the picture.
Thank you so much, Jamie.
Great. So, and I think we have another question on the from the attendees as well. So given the major advancements in data in melanoma, have there been any insights from these data sets as to which patient profile, specifically in biomarker-driven or not, benefit the most, and where are the gaps? And if you could take that question on.
Sure. Very hot topic. So this is relevant, I would say, on two different areas in melanoma. One is in first line and one is in adjuvant. So starting with first line, our options today are PD-1 monotherapy. We've got a nivo-ipi combination. We have BRAF/MEK therapy. And of course, the choice is first informed by BRAF status. So that's the first sort of very clear-cut biomarker that is very well entrenched today. And then you have some patient-specific considerations to choose between IO monotherapy versus combination. And what we're expecting to see as PD-1 LAG-3 is added to that mix, we're hoping that this will provide a strong efficacy coupled with better safety than the nivo-ipi combination that's available today. And so we could see patients choosing the LAG-3 option that previously would have received either the PD-1 monotherapy or the nivo-ipi based on their particular circumstances. Then when it comes to adjuvant, the question of patient profile might be even more important here because some of these patients are going to be cured and never need additional disease, but some of them will relapse and pass away from their condition. Although the current studies have an all-comers approach, I would say there's been continued interest from the physician community to better understand which patients benefit the most from adjuvant therapy. But that's something that, to your question, unfortunately, we don't have from the current data sets, but we're watching for as the field evolves.
Amazing. Thank you. Thank you so much, Andrew, Sarah, Jamie, and Anne for such an amazing overview. Again, attendees, please feel free to use the chat or Q&A feature to pose the question. Now we are going to move to the next set of tumor type.
Now I actually have your permission to actually go into
Yes,
breast
please.
cancer then. With that, Joel Tamayo, if you're ready, we can step right in. Again, Q&A, feel free to send in questions. And as some of you have been texting me as well as Mahim, happy to receive questions via text. And I'm getting some protein degraders and some other questions. So please keep them coming. We'll pick them up throughout the course of this session. Joel, back to you.
Great, thanks, Hafiz. I just want to make sure you guys can hear me. Thumbs up. Okay, great. So before I discuss the exciting developments that we saw in breast cancer, I thought it would be important to highlight that despite all the advances we saw in 2021, breast cancer surpassed lung cancer last year to become the most commonly diagnosed cancer worldwide. So while we've seen a lot of progress, there's a lot of work that we still need to do to address these patients. You know, that said, at the beginning of this year, or of 2021, the FDA converted the accelerated approval of sacituzumab govitecan in triple-negative breast cancer to a full approval based on the ASCENT trial, making it the first antibody-drug conjugate approved in triple-negative breast cancer. And importantly, the approval was granted an expanded indication that includes second-line patients whose options have been previously been limited to chemotherapy. Then in July, we saw checkpoint inhibitors move into the early setting for high-risk triple-negative breast cancer patients with stage 2 and stage 3 disease. And specifically, the approval was for pembrolizumab in combination with chemotherapy as neoadjuvant therapy, followed by pembrolizumab as monotherapy in the adjuvant setting. We also saw very impressive results from the DESTINY-Breast03 trial, which was a head-to-head study evaluating trastuzumab deruxtecan versus T-DM1 in HER2-positive breast cancer, making it the new standard of care for these patients. And finally, we saw the approval of abemaciclib as adjuvant therapy in high-risk patients with HR-positive disease, making it the only CDK4/6 inhibitor approved in this setting and the first drug approved in the early HR-positive setting in over 15 years. As we head into 2022, we have a number of exciting events that we're expecting. In the first half of this year, we're expecting the first PARP inhibitor to be approved in the adjuvant setting for BRCA-mutant HER2-negative breast cancer patients. And then trastuzumab deruxtecan is also expected to be approved based on the DESTINY-Breast03 results I mentioned earlier. We're also expecting to see data from TDXD and DESTINY-Breast04, which, if positive, will create a new subtype of breast cancer specific to HER2-low expressors. And then we also expect to see data from the TROPiCS-02 study, which is evaluating sacituzumab in pretreated HR-positive breast cancer. And depending on the data, we could see ADCs available to patients with HR-positive disease as well. In the second half of this year, we could potentially see the first approval of an oral SERD based on the EMERALD trial that was presented at SABCS last year. And we expect to see data from some other oral SERDs, which could inform whether these oral SERDs will have a class effect or if there is some differentiation here. And then finally, we're expecting some data from the Capitello 290 trial evaluating the AKT inhibitor capivasertib in triple-negative breast cancer, which, if positive, would be the only AKT inhibitor to show a benefit in this setting. In terms of the trends that we've seen, it's clear that ADCs are gaining a prominent role in the breast cancer treatment paradigm, with approvals in triple-negative breast cancer and HER2-positive breast cancers, and soon potentially HER2-low, and eventually even HR-positive breast cancer. Moreover, we see that ADCs are beginning to be evaluated in earlier settings, with trials like Saskia and DESTINY-Breast05 looking at the potential of ADCs in patients with residual disease after surgery. And as we saw on the last slide, there are a number of oral SERDs that are currently in clinical development. And since these have the promise of overcoming endocrine resistance while providing the convenience of oral dosing, we expect that these could potentially become the standard endocrine therapy backbone in the future, especially as they are evaluated in combination with targeted therapies like CDK4/6 inhibitors. Finally, while we didn't talk about this too much, as treatments move into the earlier settings in breast cancer, precision medicine techniques are also becoming increasingly utilized in clinical trials to determine eligibility based on ctDNA, for example, as in the ZEST trial, where ctDNA is being used to determine BRCA mutant status for treatment with a PARP inhibitor, or to determine response to treatment, as in the Saskia trial I mentioned earlier. So with that, I'd like to open it up for any questions.
Amazing. Thank you so much, Joel. And as always, there is a lot going on in breast cancer. So I appreciate you summarizing some of the key trends and implications. I heard a term which is a little new to me, and I'm sure it's a little new to some of the attendees as well. You talked about HER2 low. So we have heard about HER2 positive, we have heard about triple negative. Can you elaborate a little bit how HER2 low subtype is defined, and how should we think about it in terms of breast cancer subtypes?
Yeah, I mean, it's an interesting question. And as you know, the current model for breast cancer subtypes is based on the presence of specific receptors, namely the estrogen, progesterone, and HER2 receptors. And these have been very useful for making many clinical decisions. But the challenge to this model has always been, how much HER2, for example, do you need to have to be called HER2 positive versus HER2 negative? And so with the DESTINY-Breast04 trial, which is what is essentially creating the HER2 low space, what we see is that in some patients who would have previously been considered to be HER2 negative may now be classified as HER2 low instead, and will have the option then to be treated with a HER2 targeted agent. Again, that depends on the results of the trial. So I should say HER2 low won't be distinct in the way that HR positive and HER2 positive and triple negative are. HER2 low expressors can also be either hormone receptor positive or triple negative or hormone receptor negative, though I think the prevalence is higher in patients with HR positive disease. And so essentially what I think will happen is that it will introduce a novel target for each of those subtypes, so that HR positive patients who are traditionally treated with things like ER targeting agents or PI3 kinase inhibitors, or triple negative breast cancer patients who will either receive checkpoint inhibitors or TROP2 ADCs, will now also have the option to be treated with a HER2 targeted agent, at least in later lines of therapy for now, since that's where DESTINY-Breast04 is limited to. But certainly it will be interesting to see how this advances.
Yeah, no, absolutely. Thank you, Joe.
Great.
Any other question?
If you have time, if I could quickly ask about precision meds, given breast cancer is such an amazing space with subpopulation and phenotyping of patients, where do you see precision meds coming in and being used in the future of breast cancer as a paradigm?
Yeah, I mean, so we already see some precision medicine techniques being used, at least in the HR positive setting, to determine ESR1 mutations, for example, and this can help to determine resistance to endocrine therapies. We talked about that briefly when we discussed the oral SERDs and their value proposition. At SABCS this year, we saw really interesting data from the PADA-1 trial presented by Dr. Bidard, where they used serial ctDNA analyses to identify when ESR1 mutations emerge, so that they could switch essentially the endocrine therapy backbone. And what they showed pretty elegantly was a striking doubling of the PFS when you switch to something like fulvestrant in those patients. So we're certainly seeing uses like that advancing in the clinic, and it'll be interesting to see how that plays out. I mean, ctDNA is also being used in trials to determine eligibility for treatment, for example, with alpelisib. That said, I think just given the challenges with sensitivity and specificity, it seems like tissue biopsy will likely remain the standard for most diagnostic assessments in breast cancer. But it is a trend that we're seeing increasing, and certainly worth highlighting, given that it is being increasingly used in clinical trials and including pivotal trials.
Awesome. Thank you so much, Joel. And I know there are a couple of questions coming in the chat as well, and we'll take them after we finish this set of 3 tumor types. But I really wanted to call out one question which we are very, very interested in as well. The question from the attendee is, are the new IO agents going to close the gap with regards to innate resistance? And of course, this is a key unmet need, especially since checkpoint inhibitors are now getting incorporated in treatment paradigm more and more. The patients who have primary resistance, as well as to a certain extent who have secondary resistance, will become a high unmet need. And we can do another panel on this as well, and we'll follow up with the attendee as well.
I was just going to say that, the team here at the table across the various geography, we actually have been calling this IO 3.0, and then the team has been investing heavily in trying to understand the new delta as the next wave comes in. And those are the exact topics we're addressing. So maybe in 6 weeks' time, we can reconvene the table for another town hall on that.
Yeah,
great.
Absolutely. And thank you so much for submitting your questions. These are really exciting questions. And I also wanted to just note that there are a couple of breast cancer questions, which we'll take after the set of these tumor types, including prostate now, and then we'll do GI, and then we'll come back and discuss some of those open questions.
Great. With that, we're jumping right in with Troy Costa, who will be walking us through prostate cancer.
Thanks, Hafiz and Mahim. 2021 held some potentially practice-changing data releases, as well as 2 impactful FDA approvals for prostate cancer. First, looking at the ASCO readout for VISION, the phase 3 trial assessing Novartis's 177 lutetium PSMA 617 in late-line mCRPC. A 0.62 OS and a 0.40 rPFS hazard ratio were shown, which is a strong efficacy benefit considering how heavily pretreated the population was, nearly 40% of whom had prior cabazitaxel exposure. At ESMO, we saw some data underscoring the importance of treating castration-sensitive disease with abiraterone-based regimen from STAMPEDE and PEACE-1, particularly in those high-risk and high-volume patients. STAMPEDE showed a 0.60 OS hazard ratio in the high-risk non-metastatic setting, while PEACE showed a 0.82 OS hazard ratio in the de novo metastatic castration-sensitive setting, with, of course, an even more pronounced benefit in the high-volume patients. In May and December, we saw the first and second commercial approvals of PSMA PET agents, respectively. And as Pylarify and Illuccix ramp up, it's very likely that there will be some impact to both staging and treatment eligibility. For this year, you can see that we are anticipating the potential of a lot of exciting data and approvals. Sticking to the highlights, at ASCO GU, we are anticipating to see the first phase 3 data on PARP NHA combos in all comers, as well as phase 3 data on an NHA chemo combination. There's also the potential for data on IO combos and NHAs moving earlier into lines of therapy later on in the year. As far as approvals go, Novartis has messaged 177 lutetium PSMA approval in the post-NHA, post-taxane mCRPC setting in both the US and the EU this half, which will be a very exciting development for prostate cancer patients. Based on press releases indicating primary endpoints were met, we are also anticipating the approval of PARP NHA combos and NHA chemo combinations, though ASCO GU data may give us more insight there. Pending data readout for OS, there's also the potential for the approval of ipatasertib and abiraterone, which would be the first AKT combo in mCRPC. Looking at key trends seen over the past year and into the coming year, respectively, we would be looking at NGI, so next generation imaging, specifically PSMA PET CT, which is anticipated to become part of the standard of care. There are, of course, several hurdles, including the fact that the commercially approved agents have only very recently been launched, as well as logistics and reimbursement that may impede how quickly this becomes part of the standard of care. Next generation imaging also presents some unanswered questions, primarily that we don't know exactly how upstaging patients based on more sensitive imaging will actually impact treatment patterns and subsequently survival outcomes. Additionally, established MOAs are expected to continue moving into earlier lines of therapy, while novel mechanisms of action are introduced into late-line MCRPC. Specifically, as sort of previewed earlier, NHA-based regimens are moving into earlier lines of therapy, either in chemo combinations in the metastatic castration-sensitive setting or into the high-risk non-metastatic setting, potentially with or without ADT. Novel MOAs, including, of course, PSMA radioligand therapy, as well as potentially AKT and IO combinations, are anticipated to enter MCRPC over the coming year and into the next year. Finally, there is a trend of targeted therapies with established benefit in biomarker-selected populations being assessed in all-comers trials when in combination. So pharmaceutical companies are looking for combinations with potential synergy, such that all-comers populations will still benefit. While we do know that early testing is important for treatment selection and sequencing, particularly considering targeted monotherapies are currently only approved in biomarker-selected populations, it will be very interesting as trials read out to see if there are, in fact, synergistic effects that will benefit all patients. So with that, I will open for any questions.
Say that again.
Wow, this is amazing. Thank you so much, Troy. It seemed, I think I saw at least 5 different kinds of targets and modalities currently with different permutation and combination going to read out soon in this year. There is NHA, there is PSMA, there is PARP inhibitor, AKT inhibitor, even checkpoint inhibitor as well. First, could you just give us a brief overview of what radioligand therapy is? And then can you also answer a question around how do you think PSMA RLTs could impact prostate cancer management?
Yeah, of course, happy to. So PSMA radioligand therapies, it's really interesting science. It's effectively an antibody that targets PSMA, which is a prostate-specific membrane antigen, and it's conjugated to a radioisotope, which will emit either beta or alpha radiation. The PSMA radioligand therapy of interest is 177 lutetium. It's Novartis's agent. That's the most likely one to get approved this half. And it is a beta emitter, so it's 177 lutetium. It binds to cells expressing PSMA, which is primarily overexpressed in prostate cells. And therefore, it will target the tumors, whether they are located in the bone, the viscera, or at the primary tumor site. So in terms of how they will actually impact prostate cancer management, as of right now, prostate cancer is primarily managed by urologists and medical oncologists as the treatment journey progresses. Although, of course, radiation oncologists, nuclear medicine physicians, and a variety of other specialties are involved. But the primary managers of care are urologists and med-oncs. As this therapy becomes approved, it's very likely that nuclear medicine physicians and radiation oncologists will play a greater role in managing these patients and administering treatment. And this will sort of bring in the factors of interdisciplinary teams managing the patient throughout the course of their treatment journey. This will also likely increase PSMA PET uptake, as well as giving a novel mechanism of action, which should help to combat issues around resistance to docetaxel and AR inhibitors.
Awesome. Thank you so much, Troy. Maybe we should move on to that.
Yeah, do we have time, Mahim, for one more question?
Oh, yeah, sure, please.
I don't know if you guys have a latest on this. Are there other emerging PSMA-directed therapies of interest, Troy?
Yes, so beyond other alpha and beta emitters that are PSMA radioligand therapies, there's also PSMA-directed CAR Ts. There are even PSMA-directed bispecifics, trispecifics, and as well as a slew of non-PSMA-directed MOAs that are coming out. I believe at ASCO GU, there should be readouts on PSMA-directed bispecifics and CAR Ts, mostly early data, but it should be really interesting to see what it looks like in terms of actual clinical performance.
Troy, thank you, Troy. We're going to answer. We have a community oncologist joining us. He has a question on breast. So, Joel, if you can quickly address the question.
Yeah, let me just phrase the question so that Joel has a question. Okay. So the question is, since breast cancer is a mosaic disease, are there any studies cooking for HER2 patients with CDK4/6 agent? That's very interesting because CDK4/6 agent have transformed HR positive. So, Joel, do you want to address that question?
Yeah, I mean, that's a great question. I think we are seeing it more in larger trials. The largest trial I can think of now that is looking at CDK4/6 inhibitors in patients with HER2 positive, HR positive disease is the Monarch-HER study, which is in the metastatic setting, showing an improvement in that case in PFS. There's another study, the Patina study, that's looking at a similar CDK4/6 with HER2 targeted agents, again in the metastatic setting. So I think it's something that, we've seen some promising data, and it's something for sure that I'm sure we'll continue to see advance earlier as well.
Thank you so much for the question. That's really interesting.
And thank you for joining, Doctor. With that, we are moving on to Dr. Jidvani on GI.
Thank you very much. 2021 really saw compelling data evaluating checkpoint inhibitors in frontline gastroesophageal cancer and hepatobiliary carcinoma. First, nivolumab and chemotherapy was approved as a first-line therapy in patients with gastric cancer, gastroesophageal junction cancer, and esophageal adenocarcinoma based on the CheckMate 649 trial. A similar outcome was observed in the companion CheckMate 648 trial that supports the use of nivolumab and chemotherapy in PD-L1 positive esophageal cancer with squamous histology. The PD-L1 expression thresholds defining ideal patient suitability in practice will be certainly an evolving discussion. Turning to HER2 positive gastric and GEJ cancer, which accounts for about 20% of advanced disease, the approval of first-line pembrolizumab trastuzumab chemo combination was based on interim data through the FDA's real-time oncology review pilot program. So here, the potentiation of first-line trastuzumab with a checkpoint inhibitor led to deeper and more durable responses than trastuzumab chemo alone. In the HER2 positive relapse setting of gastric cancer, trastuzumab deruxtecan, an anti-HER2 ADC that's been mentioned today, was approved based on the phase 2 DESTINY-Gastric01 study, which was conducted in Asia, but with ongoing supporting trials, including Western populations. In frontline HCC, the Himalaya trial met the primary endpoint of overall survival when patients received a priming dose of anti-CTLA therapy in combination with a regular schedule of anti-PD-L1, durvalumab, that was presented at ASCO GI just last week. And so with Atezo plus Bev already approved in the first line setting, options have really expanded from the TKI sorafenib to IO plus TKI to IO-IO. And so the sequencing of treatment in this space is now an open question and may be determined by various patient characteristics such as sensitivity to anti-angiogenics and performance status. In BRAF mutant colorectal cancer, patients with the V600E mutation progressing on first or second line therapy saw significant OS and PFS benefit from the targeted doublet of cetuximab and encorafenib. Notably, this is a chemo-free targeted therapy and may form the backbone of future combinations in this population, including with nivolumab. Looking ahead to 2022, we could see the approval of the durva-tremi-stride combination, along with revised pembro plus lenvatinib data in frontline HCC, expanding treatment options and TKI sequencing decisions. Updated data from the pivotal phase 2 Mountaineer trial evaluating dual anti-HER2 combination wild-type metastatic CRC could lead to accelerated approval. Although in second-line pancreatic cancer, the pivotal phase 3 trial evaluating the metallotropic drug ariaspase was negative, phase 1 data for the drug in frontline could signal potential efficacy when used earlier and with platinum chemo. Among the trends we're seeing in GI, we're seeing an expansion of biomarker-driven therapy options, including checkpoint inhibitor combinations in the frontline setting and anti-HER2 therapy in frontline and second line, where existing options in the relapse refractory setting are limited. Another important trend is the increasing use of liquid biopsy to detect circulating tumor DNA. And this has benefits in the adjuvant setting to guide choice of therapy based on the presence or absence of minimum residual disease following surgery. And in the metastatic setting, post-frontline therapy, as tumors and clones may undergo mutational changes, reversing resistance, and thus rendering them susceptible once again to targeted therapy. Finally, aspects such as IO and TKI re-challenge have emerged. A major question will be the sequencing of therapies based on biomarker status. And a major implication of these successes will be on the control arms that are accepted in future studies now that checkpoint inhibitor-based regimens will be included in standard of care. And I'd like to turn it over to any questions from the audience.
Yeah, good.
Okay, thank you, Dr. Zubani. This is great. And obviously, we were very excited to see some of the PANC data that came out over the weekend. In terms of pancreatic, what emerging data or MOA that are of high potential, and what are some of the next hopes in pancreatic?
That's a great question, Hafiz. Notably, pancreatic cancer has been a pretty tough nut to crack. A lot of trial failures, despite some early successes in phase 1, phase 2. But I'd say in advanced pancreatic cancer, the CD40 pathway has really been a target of interest. And the goal here is to overcome the immunosuppressive tumor microenvironment through the activation of antigen-presenting cells. So including dendritic cells, B cells, to essentially amplify antigen uptake and presentation, ultimately coordinating a more powerful immune response, including in combination with checkpoint inhibitors that would otherwise be ineffective in a cold tumor such as pancreatic. And so a number of CD40 agonist monoclonal antibodies are in development, and at ASCO GI we saw that data that supported the hypothesis that non-fucosylated antibodies impart greater antibody-dependent cellular cytotoxicity, or ADCC, essentially greater anti-tumor activity. And so this was an exciting finding. I should also mention that the target Claudin 18.2 is a hot topic in gastroesophageal as well as pancreatic, both tumors where it is highly expressed. Additionally, I should also mention that cell therapy is very much entering the GI space, including a trimodal NK cell therapy from ImmunityBio in pancreatic cancer, as well as TCR-T therapy in esophageal cancer from Adaptimmune. Both of these trials, again, were highlighted at ASCO GI last week.
Awesome, great. I think you have already mentioned some of the insights from the recently finished conference ASCO GI, but are there any other treatment-changing data that read out at ASCO GI that you want to highlight?
Well, I think I just want to go back to that point about the Himalaya trial. This was really compelling in first-line HCC as it leveraged the temporal synergism of anti-CTLA and anti-PD-L1 therapy. So in this trial, an initial pulse of tremelimumab primed the T cells to then better be able to respond to regular durvalumab treatment. So this is now known as the STRIDE regimen, or the T300 plus D regimen. So superiority in this trial versus standard of care sorafenib was achieved with the IO doublet, and this conferred a 3-year overall survival of 31%, associated with that long tail of the OS curve that is characteristic of a delayed but persistent IO response. Additionally, from this trial, durva monotherapy was non-inferior to standard of care sorafenib. So the outcome of this trial is potentially 2 additional treatment options in frontline: durva tremi, which is free of anti-angiogenic therapy, as well as durva monotherapy for patients who are not suitable for combination therapy.
Awesome. So Rohan, there is a question from the attendee in a chat. So the question is, how is the KRAS target therapy applicable in pancreatic cancer?
That's a fantastic question, and I think we saw at ASCO GI some really great data from Crystal-1, a data update from Crystal-1, showing some very strong responses in pancreatic, but GI overall. And I'd say that the KRAS space is certainly looking forward to KRAS G12D inhibitors, and there are a couple on the market, but certainly the G12, I should say the G12C, which was presented at Crystal in Crystal-1, but G12D and G12V are the KRAS mutations that are probably more relevant to pancreatic cancer and colorectal cancer. So it'll be really exciting to see further data from those trials. But just to mention that they're in colorectal cancer, the Crystal-10 trial is ongoing, and that will explore a KRAS G12C inhibitor in combination with cetuximab in second line.
Awesome. So thank you so much for the question. Definitely a very active field within pancreatic cancer as well. I would like to invite Troy back as a panelist now from prostate, and I know Joel from who just summarized breast cancer is in the big table sitting with us. So I would just like to get each of your sort of 2 cents on what are some of the emerging MOAs or class of drugs that you are really excited about in your tumor type. So I would like to start with, let's say, Troy, do you want to start the discussion?
Happy to jump in. So there's a lot of exciting stuff going down in prostate cancer. I think that it'll be most interesting to see IO entrants, given that to date, IO monotherapy has a slight history of lackluster responses, but IO combination therapies in smaller studies have been showing some promise. So it'll be really interesting to see some IO data as these IO combos read out in these larger phase 3 studies later this year and early next. And this also kind of ties into some of those PSMA-directed therapies earlier when it comes to the CAR T, given that it'll be really interesting to see how CAR T can perform in solid tumors, specifically prostate cancer, and that preliminary data has shown some efficacy, as well as bispecifics are pretty interesting.
Awesome, thank you. Joel, do you want to go next?
Yeah, I mean, we didn't really talk about cell therapies at all, since there aren't really any cell therapies in late stages of development. But I guess I had to think about some of the new modalities that we're seeing in breast cancer. As cell therapies are being evaluated in solid tumors in general, there are a few now that we are seeing in early phase trials in terms of allogenic cell therapies. I think recently we saw that a small company is developing a CAR-M therapy, CAR macrophage therapy. And that's being developed in combination with a checkpoint inhibitor in HER2 positive tumors. And we've seen some preclinical data to suggest that there's a potential additive effect there in combination with PD-L1 blockade. So I think that's some of the modalities that we're going to see, I think, in the future.
Awesome, thank you so much, Joel. Rohan, from GI perspective, anything you want to share with the attendees in terms of next wave within GI?
Yeah, I think one of the conversations that I think we're having right now overall is ctDNA liquid biopsy. And so just to point out the different ways in which it can be used, of course, in a setting of early disease, ctDNA quantitates the level of minimal residual disease following surgical resection. So for MRD positive patients, oncologists may recommend the use of a more aggressive adjuvant therapy. And conversely, with lower ctDNA threshold, de-escalation of adjuvant therapy could be the more appropriate decision to eliminate undue treatment burden. And then following surgery, patients may look phenotypically similar, but may have some underlying biological component predictive of risk of relapse. So ctDNA analysis can really distinguish between these cases at a cellular level. So the monitoring aspect is certainly important. And in the metastatic setting, applying a targeted therapy strategy based on actionable mutations requires tissue for testing, NGS point mutation. So here, liquid biopsy is useful as it's non-invasive, doesn't require surgical biopsy, and has a very quick turnaround time. So it's important when you need to make decisions very quickly on frontline, as well as when you're re-challenging with anti-HER2 or anti-EGFR therapy.
Awesome, that's great. And we'll sort of revisit some of these discussion points around ctDNA and liquid biopsy in our precision medicine session or sub-session as well. Now moving on to the next.
Okay, we took enough time, so shall we jump into hematology? So with hematology, first comes myeloma, and who else rather than Dr. Wajia Ali walking us through some of the myeloma. Also, we have, Mohammed Khan here joining us from myeloma, who's walking us through some of the 2021 events as well as catalyst in 2022.
Great, thank you, Hafiz. So starting off with the key events for myeloma in 2021, the beginning of this past year started off with two exciting approvals, Abecma or idacaptogene viclucil, and additional approval for Sarclisa or isatuximab with carfilzomib and dexamethasone. Abecma was the first CAR T approval for multiple myeloma, and the approval of Isa-CAR-Dex follows the approval of Isa-POM-Dex for a similar setting earlier in March of 2020. Q2 saw a lot of updates for Janssen, mostly around ciltacabtagene autoleucel and teclistamab, their upcoming CAR T and bispecific that have cleared many regulatory hurdles and are on their way to approval soon. Q3 also saw the approval of Darzalex Faspro for Janssen in July of 2021, the subcutaneous formulation of the already approved Darzalex, which was given IV. And also in Q3, Pepaxto or melflufen was withdrawn after communication by the FDA. And this last quarter, the biggest developments came from the ASH meeting. Many of the myeloma abstracts centered around CAR T and bispecifics, though there was attention given to ADCs like belantamab mafodotin and other drug classes like SINEs, including selinexor. As for what we hope to find in 2022, I think the most anticipated event of the first half is going to be the approval of cilta-cel, or Carvykti as it will soon be known, whose PDUFA date is due on February 28th. And also, since the BLA was submitted for teclistamab in this past December, and the data is rather strong, we might see a teclistamab approval in the second half of the year. And this would be the first bispecific to be approved for myeloma. In terms of data, we can expect to see there are many ongoing daratumumab trials, the most interesting of which are in transplant-ineligible disease, and also smoldering multiple myeloma, which is a precursor to multiple myeloma. We're also looking forward to seeing isatuximab data in a first-line quadruplet regimen, venetoclax and T1114 mutation patients in the CANOVA trial, and other bispecifics beyond teclistamab, of which there are many. Summarizing some of the trends that myeloma can expect to see as a whole, the three trends that we can look forward to this upcoming year are the emergence of bispecifics as a standard drug class, the standardization of quadruplet regimens in newly diagnosed multiple myeloma, and the inevitable role of CAR T therapies. Bispecifics are in a good position to be approved soon and are more convenient to administer than autologous CAR T. Quadruplets with daratumumab are becoming more of the norm in newly diagnosed disease versus triplets. And lastly, autologous CAR T is certainly here to stay, seeing as ide-cel and cilta-cel provide such great and durable responses, and there are others in the pipeline undergoing clinical trials at present. And with that, I thank everyone for their attention, and I'll welcome any questions.
So now, Mohammed, can you give a quick overview of current states of bispecifics and CAR T in myeloma, please?
Yeah, definitely. I'm glad this was asked because I didn't get the chance to sort of elaborate on this due to time. But the approval of Abecma was extremely pivotal in myeloma, as it was the first CAR T for approval, though we had seen previous CAR Ts in other heme malignancies. We're anticipating cilta-cel to be approved as well in early 2022, and overall clinical trials are in place to bring CAR T to earlier lines, even as a first-line therapy like CARTITUDE-5. This has all been for autologous CAR T, though, and there are assets in development for allogeneic CAR T, and we can hope to see promising late-stage data at future readouts. As for bispecifics, there are actually a lot of assets in development, most notably teclistamab, which was submitted to the FDA in late December. We also have things like elranatamab in late-stage development, and also targeting BCMA like teclistamab, while there are other non-BCMA bispecifics like cebostamab and talquetamab. These are currently indicated for later line populations as well, but once we see approvals, it's not unlikely that they will also try to enter earlier lines, just like autologous CAR T.
Thank you, Mohammed, and great job picking up myeloma in such a short amount of time. Beyond bispecifics and CAR T, what are some of the other classes or MOAs that you're excited about?
Yeah, that's a great question. Besides CAR T and bispecifics, the myeloma space is also exploring tri-specific antibodies that also bind to albumin in addition to BCMA to increase in vivo retention, and also CAR T therapies beyond just BCMA. Like I mentioned, we see non-BCMA bispecifics like FcRH5 for cebostamab or GPRC5D for talquetamab, and these are also targets for developmental CAR T therapies. We also have one approved ADC in myeloma, which is belantamab mafodotin, but at present it causes ocular events in most patients. So there are developmental studies that seek to rectify this. So maybe at a future looking ahead, I'll be talking about all these drugs with great data. So we'll stay tuned.
Awesome. Great. Thank you, Mohammed. Do we have time for more questions? Do we need to jump into lymphoma and leukemia?
Let's jump into the next tumor type and we
Great.
can take some questions at the end.
Great. With that, Wajih Ali.
Take a second as Wajia is setting up.
Okay, great. And keep your questions coming. If there are questions as they're coming in, if we don't get to you in this next 20 minutes, we'll absolutely be responding back to you via email or other ways today. Okay, Wajia.
So much, Hafiz. So I'm going to be covering some of the top highlights from 2021 across DLBCL, FL, CLL, AML, and MDS. Starting off with CLL, we saw the positive readout for the ELEVATE-RR study. This was essentially the first time that one BTK inhibitor was compared against another, and this trial proved non-inferiority. We also saw the approval of umbralisib in February 2021 for follicular as well as marginal zone lymphoma. This was a next generation PI3K inhibitor. So this forms a new drug in the class of drugs that's already been approved and is used as a standard therapy. Moving on to the MDS space, it was a mixed bag because we saw some good regulatory progress for the Ven-Aza combination where it got a BTD. And this would be a standard treatment for previously untreated intermediate high or very high risk MDS if approved. While at the same time, we also saw two major trials either reading out negative or having FDA hold placed on them. So it was a mixed bag for MDS. And Q2, we also saw some long-term data of CLL trials. What these trials demonstrated was that the standard treatment patterns that have been set for the last couple of years in CLL were solidified. There was more evidence to support what was already happening, whether it was the use of venetoclax plus obinutuzumab or the use of new BTK inhibitors or essentially treatment options for elderly patients in the form of ibrutinib and venetoclax. In Q3, we saw a big development in DLBCL where we saw polatuzumab's POLARIX trial reading out positive. So this essentially would enable a shift in treatment from the R-CHOP standard to a POLA-R-CHP standard regimen. And this would be perhaps a new first-line standard of care. Lastly, in Q4, what we saw was a big development for the follicular space where we saw the first bispecific CD20 antibody being filed for approval for the third line FL space. And this essentially forms a new treatment approach for this section of patients. And lastly, while we speak about these clinical trial developments, it's also important to know how the disease is being classified. So we saw an addition of gene mutations into the IPSS staging system for MDS, and this will perhaps form the basis of some of the newer trials and treatment modalities. Moving on to what we expect in 2022 for some of these tumor types. Again, a lot of exciting changes and we've just tried to capture some of the high insights, and I'm sure we can find more developments if we dig deeper into each of these tumor types. Starting off with MDS, we will perhaps see the approval of Ven plus Aza, creating a new standard of care. We'll also see a lot of changes to perhaps the treatment of DLBCL. And these changes will follow through into different lines of therapy. So we can see the first line evolving with POLA's introduction. We also saw the second line actually having some data to prove that cell therapy has a place. And this was essentially perhaps form a new treatment option for certain patients who are selected for it. The bispecifics, we saw in the last slide that there is the first filing that's done, but we also look forward to some of the data from these trials, and that would form the basis of a new standard in the third line setting. We're also excited to look at the AML space and the MDS space where agents such as menin inhibitors or CD47 inhibitors would probably see some role. And lastly, to round it off, we have two other developments in CLL, looking at ibrutinib plus venetoclax in the first line setting that would perhaps be a good standard option. And lastly, cell therapy is key or essential in the treatment of lymphoma. We've seen approvals, we've seen line movements, we've also seen data in new segments. But what we expect to see in 2022 is essentially the next level of cell therapy, whether it's allogenic, whether it's NK cell-based, gamma delta, viral. And we're looking forward to some data that forms the proof of concept for these agents. And to summarize the broad trends within the heme space, what we're looking forward to is essentially the entry of bispecifics. So 2022 is perhaps going to be the year of bispecifics in heme. We will look forward to data, and we will also perhaps look forward to some regulatory decisions on this class of drugs in the DLBCL and FL space mainly. We'll also perhaps look at MDS finally having some viable treatment option. It's a very heterogeneous disease. It's always suffered from unavailability of the right targets against which drugs can be developed. And hopefully in 2022, we'll see some progress amongst the setbacks that we've seen in 2021. Finally, cell therapy, the first wave of cell therapy essentially was transformational, but it also had a lot of logistical as well as administration hurdles, and that was also in part due to the pandemic. And we're hoping that the next generation of cell therapy would be more suitable for patients, more easily administered, and of course have the high efficacy and the curability potential that the cell therapy class as a whole promises.
Great. In the interest of time, if I can ask one question, Mahim. So what is the role of BTK inhibitors in CLL, Wajia, before we head into precision meds?
Yeah, so BTK inhibitors have been the backbone. It started with the first generation, and these formed a new targeted therapy approach. But unfortunately, they had some off-target effects. So the next generation, which is zanubrutinib or alakibrutinib, were developed to counter those off-target effects and increase the efficacy and safety profile. But now there's a third generation that is coming forward, which is looking at specific mutations that are associated with resistance. So it's essentially it's going to enhance the efficacy line after line to ensure that patients who benefit from BTK inhibitors are able to get the best option and are able to take the drugs for a long period of time.
Awesome. Thank you so much, Wajia. Let's move on to the next.
Yeah, absolutely. And so we'll give a quick second for Dr. Kane to actually settle into precision meds. And precision meds, as you can see, goes across, and tumor agnostics will have initial chat, and we will tie back and bolster back to some of the discussion Dr. Anshari was having on liquid biopsy, and so the next follow-on discussion in a town hall in a couple of weeks as well. Dr. Kane, let us know when you're ready.
In the meantime, we just wanted to say we appreciate the questions being posed in the chat. We'll try to address it at the end, and if not, we'll follow up with our perspective with the individual attendees as well. All right, over to you, Charlotte, whenever you're ready.
Hi, thank you very much, Hafiz and Mahim. And so we're going to wrap up today's year in review with the broad topic of precision medicine. And so which really ties in nicely to the indications that we've discussed already today. A few key events that occurred in 2021 during and around large medical conferences. At AACR, we saw multiple data presentations using liquid biopsy for monitoring patients with after surgery or adjuvant therapy. At ASCO, GRAIL presented the first data set from their interventional Pathfinder study using Gallery, the first commercially available multi-cancer early detection test. And then in the topic of biomarker testing, both at ASCO and ASH, we saw real-world data sets showing low testing rates among US community oncologists in both lung cancer and chronic lymphocytic leukemia, where over 50% of patients do not get tested for NCCN-recommended biomarkers and subsequently don't necessarily receive the appropriate therapy associated with those biomarkers. And so in the next couple of slides, we'll be reviewing how some of these key events impact our trends for 2022. So first, we do expect to see an increased use of liquid biopsy, particularly for monitoring in the adjuvant setting. We've seen large data sets generated across multiple solid tumor types, and we do expect that this will ultimately impact patient outcomes because we will be able to potentially have earlier intervention based on detection of molecular relapse with these assays. As early detection cancer tests arrive to market, we do expect to see a stage shift in cancer stage diagnosis, having patients with earlier stage disease, which have historically been shown to have improved outcomes. In light of this, we also expect a spur in new clinical drug development in order to be able to treat these patients and provide them with potential targeted therapies or immunotherapies. And finally, again, in the liquid biopsy space, we did see in 2021 and expect the trend to continue, exploring the use of ctDNA as a surrogate for tumor burden in solid tumors, based on the presence or absence of ctDNA. We've seen novel study designs that are incorporating this tool in order to properly segment patients and make informed treatment decisions. As my colleague Rohan mentioned, CRC is really a key study, a key case where we're seeing this being used and for patients receiving different treatment regimens based on the results from the ctDNA assay. As we move to more and more targeted and precision medicine approaches for these patients, we can imagine this tool being incorporated to clinical practice and properly identify which patients will benefit from an adjuvant therapy or which may benefit from a treatment-free observation period. So to be continued. As we move on to another theme within precision medicine, being biomarker testing. As we previously mentioned, we saw 2 key presentations in 2021 showing those low testing rates both in lung cancer and in CLL. A few barriers were really highlighted, showing that access to NGS and physician education about NGS are some of the key points that we can start addressing to improve these testing rates and patient outcomes. Another key element in biomarker testing, as targeted therapies and immunotherapies move to the frontline setting in metastatic disease, we've noticed really this increasing need to better understand the mutational landscape upon relapse. By testing patients when they relapse, there's an opportunity to identify novel biomarkers, acquired resistance biomarkers that may make these patients eligible for targeted therapies or immunotherapies. And finally, we'll wrap up where we expect to see an increased use for NGS within AML, testing for MRD. The European Leukemia Net published at the end of 2021 new guidelines that incorporates NGS alongside flow cytometry to monitor patients with AML for MRD. And with that, I will open it up for questions.
Thank you so much, Charlotte. So if I can start, and then I'll pass it on to Hafiz for a question as well. Are there any other early detection tests in development that use a different approach than GRAIL's and may target a different patient population?
That's an interesting question, and really early detection is a hot topic right now. We know GRAIL have their Gallery test that's now commercially available, but there are actually a lot of other early detection tests that are in the works. Some are using methylation sequencing like GRAIL, which is considered a bit the gold standard right now, but others are incorporating genomic testing, proteomics, RNA sequencing, even some are putting in the microbiome. So we'll really wait and see. We expect this field to expand pretty rapidly and exploring the different technologies that can be used for early detection.
Great. So in terms of early detection, and if we flip the coin to monitoring of the patients in the metastatic setting, right, in terms of prognosis, where do you see the value added there with precision meds?
So I think, again, a great question. I think it's really twofold in metastatic disease. In the use of liquid biopsy to monitor these patients, we can really monitor at shorter time points response to therapy, and ultimately we'll quickly identify, first of all, patients who are not responding to therapy, and those patients might be eligible for an add-on therapy or a different targeted therapy. And then we'll also be able to identify patients who do respond to therapy, and those might be eligible for a treatment-free observation period. We actually saw a very interesting study design using that type of approach at ASH.
Awesome. Thank you so much, Charlotte.
Great. Thank you, Dr. Kane. I want to thank the whole table here that has been working on this for a couple of weeks, as well as the team in Boston, team in India, and other areas as well. And thank you for the panelists for joining us today, and great questions. So Mahim and I will go back to some of the questions and provide input and feedback back via email and other ways. And as I said, loads of town halls coming. Anne is working on a few other things in precision meds with Luke and Charlotte. Mahim and I'll be back with the IO 2.0, IO 3.0 in terms of the next wave, and of course the topic of disparity in cancer care that is very close and near to our heart. So more to follow in this space. Thank you for spending your 90 minutes on a busy morning today or busy afternoon. We look forward to seeing you again soon. Thank you again.